All right, we're still continuing on with meiosis, and if you'll recall last time, this is where we left off. This is at the very end of Meiosis I, so we're about to go on to Meiosis II, and I just want to remind you that both daughter cells that result from Meiosis I are haploid, have half the number of chromosomes, and they are genetically unique from each other and from the parent cell due to crossing over. So now let's go on to Meiosis II and see how that works. The first thing that we want to note here is that Meiosis II is mitosis-like. If you understand the process of mitosis, then you will understand the process that's taking place here in Meiosis II. So here are the two cells that are coming from Meiosis I. Both are going to go through Meiosis II, and they're simply going to divide by the same mechanism as mitosis. And it's important to note that in between Meiosis I and Meiosis II, there is no interphase II that precedes Meiosis II. So that means that there is no G1S, G2 in between, and if there's no S, that means there's no DNA synthesis. And that's okay because the chromosomes are already duplicated. So there's no reason to duplicate anything because we already have duplicated chromosomes, and that leaves us something else to divide. You know, in Meiosis I, we separated the homologous pairs from each other. And in Meiosis II, now we're going to go ahead and separate the chromatids from each other, and then they will be considered unduplicated chromosomes, which is exactly the same thing that happens in mitosis. The only other thing to note here, though, is that the centrosomes and centrioles must replicate, and centrioles only in animal cells, because we have to have two centrosomes to form a spindle in Meiosis II. Other than that, because we've already learned about mitosis, you already know what happens here, so we're just going to run through the phases very briefly here just to remind you what goes on. So we're starting off with haploid cells that are genetically unique from Meiosis I. During prophase II, the nuclear envelopes break down and the centrosomes begin to form the spindle. So that's what we're seeing here. During metaphase II, the duplicated chromosomes line up along the metaphase plate. There they are, lined up. During anaphase II, the chromatids separate, just like in mitosis. But there's no reduction in chromosome number here. They've already been reduced. But we are going from duplicated chromosomes to unduplicated chromosomes, just like in mitosis. As soon as those sister chromatids separate, they're now considered unduplicated chromosomes. Then we have telophase II and cytokinesis II, and we end up with 4 daughter cells that are haploid, have half the number of chromosomes from as compared to the original parent somatic cell, and they are genetically unique not only from that parent cell, but from each other and every other gamete on Earth. Because if you'll recall, this is the purpose, producing gametes is the purpose of meiosis in sexually reproducing organisms. So each of these will become a gamete. Sort of. You'll see in a minute when we go over oogenesis, that's not entirely true. So here we're looking at spermatogenesis, which is the genesis, when it's part of a word, always means the beginning of. So it's the beginning of spermatogenesis. And this takes place in male sexually reproducing organisms. And you'll notice that we're condensing all the different phases of meiosis. So we're just looking at Meiosis I and then Meiosis II. So the somatic cell that we start out with is known as a primary spermatocyte. So that's a diploid somatic cell, and it's specialized to be the primary spermatocyte, the starting cell of spermatogenesis. And that divides through Meiosis I, and the 2 resulting daughter cells that are now genetically unique and haploid. And unfortunately in the diagram here, they're not showing that they've crossed over. They're not showing the color coding where pieces of the chromosomes have crossed over or anything, but they would be genetically unique because of that. They are, however, showing that they are genetically unique because they are receiving different members of the homologous pair from each homologous pair from this cell. So those are secondary spermatocytes, and then they undergo meiosis II. And the resulting 4 cells from Meiosis II are known as spermatids. And notice that they now contain unduplicated chromosomes. And again, in this diagram, they're not really showing that these two spermatids are different from each other and these two spermatids are different from each other because they're not showing crossing over. But crossing over would definitely make these genetically unique. The spermatids then will differentiate and become specialized spermatozoa. So the technical term for sperm is spermatozoa. We just call them sperm for short because it's a lot easier than saying spermatozoa. And they kind of look like little protozoa, right? Flagellated protozoa. And that's where the zoa comes in, because we call protozoa because they look like little one-celled animals, and spermatozoa are called spermatozoa kind of for the same reason, because they look like little one-celled organisms, animal-like organisms. And of course they are animal-like because they're the product of animal cells. So to summarize here, spermatogenesis is the process of producing haploid sperm from diploid somatic cells by the process of meiosis. Spermatogenesis begins with a somatic diploid primary spermatocyte. Meiosis I produces 2 haploid genetically unique secondary spermatocytes. Meiosis II then produces 4 haploid genetically unique spermatids, which differentiate and develop into specialized spermatozoa. And all 4 become spermatozoa. And I'm pointing that out because, as you'll see in oogenesis, it's different. So right off the bat here, you can see that oogenesis is different. And so we're going to do the same thing with this diagram we did with the spermatogenesis. Primary oocyte. And then we have secondary oocytes, but notice that one of them is greatly reduced compared to the other, and that's because of what we call asymmetric cytokinesis. The cytokinesis is not symmetric. It's not even and equal. So one of the secondary oocytes end up having much less cytoplasm than the other. And then both of those undergo meiosis II and produce an ootid, but notice it's just one ootid, and then the other ones are referred to as polar bodies. And they're called that because they emerge from one of the poles of the cell at the time of division. And then the ootid differentiates, and one ootid differentiates and develops into an ovum, or an oocyte, which is an egg. So we end up with one egg, or ovum, and 3 polar bodies that have basically sacrificed their cytoplasm to the egg, and the egg is much larger than what we see in the sperm through spermatogenesis, because the sperm all become, the spermatids all become spermatozoa, so none of them sacrifice their cytoplasm for the other, and they all end up being the same size, and they all end up being spermatozoa. But here, 3 of the cells sacrifice their cytoplasm so that the egg or the ovum can have extra cytoplasm and be extra large, because it needs all those resources to become the zygote at the time of fertilization. So again, to summarize in writing here, oogenesis is the process of producing a haploid ovum from a diploid parent cell by meiosis. It begins with a somatic diploid primary oocyte, just like in spermatogenesis. Meiosis I then produces 2 haploid genetically unique secondary oocytes, but one is greatly reduced due to asymmetrical cytokinesis, that's that one, which continues in Meiosis II, producing 3 haploid non-functional polar bodies, a total of 3, because another one comes from the other secondary oocyte through asymmetrical cytokinesis, and 1 haploid genetically unique ootid, which differentiates and develops into a specialized ovum, ready to be fertilized and become a zygote, which will then go on to develop into an embryo, and so on. So that's meiosis and the production of gametes by meiosis, both sperm and egg. So let's go ahead and take a comparative look at mitosis versus meiosis. So let's look at DNA replication first. So DNA replication occurs during interphase, and more specifically during the S phase of interphase before mitosis begins. It also occurs during interphase before meiosis begins. So that's the same. Number of divisions, in mitosis it's only one, including prophase, metaphase, anaphase, and telophase, and in meiosis it's two, each including prophase, metaphase, anaphase, and telophase, and that's why you need to designate prophase 1, metaphase 1 versus prophase 2, metaphase 2, and so forth. Synapsis of homologous chromosomes forming tetrads. Well, we didn't talk about that at all back when we were looking at mitosis, so it does not occur. But it does occur during prophase 1 of meiosis, forming tetrads, groups of 4 chromatids, and it's associated with crossing over. That's what introduces more genetic uniqueness to those cells. And that crossing over is between what we refer to as non-sister chromatids, those being chromatids across a homologous pair of chromosomes. Then the number of daughter cells in genetic composition. In mitosis, there are 2 daughter cells, each diploid, each genetically identical to the parent cell and each other. In meiosis, we have 4, each haploid, which means they have half the number of chromosomes, genetically unique from the parent cell and from each other. The role in the animal body for mitosis, that enables a multicellular adult to arise from a zygote. So in other words, just like us, we start out as a zygote, the one cell that results from the sperm fertilizing the egg, and then that zygote starts to divide into an embryo, which develops into a fetus, which develops into eventually a baby, and then a multicellular adult. So that's all by the process of mitosis. So from the zygote all the way to what you are right now and what I am right now was all produced through mitosis. It also produces cells for growth. So I'm not growing right now, but you are, and that's how you're growing. You're adding more cells to your body. And also tissue repair. If you hurt yourself, that's how you heal. Also note this includes replacement, because you're constantly losing cells, and those are being replaced by new cells, and those new cells arise by the process of mitosis. When it comes to meiosis, we're producing gametes. That's really the only purpose of meiosis in sexually reproducing organisms, is to produce gametes. Reduces the number of chromosomes by half and introduces genetic variation among the gametes, and that's extremely important, and really the reason that sexual reproduction exists is to introduce variation into populations, and that's why meiosis exists. And that concludes our review of meiosis.